Okay, apologies once again. So we were just talking about gene splicing, which is a more complicated technique, but it is interesting and it does involve replicating DNA. So gene splicing basically means slicing. If you think it's slicing something or splitting something, that's what it means. So you're looking for a specific gene and you're going to split it, or you're going to slice it so that you can insert another segment of DNA within that gene. Or you can look at the whole genome and you can splice part of it, and you can take a gene from something else and insert it into that genome. Okay, so that's what splicing means. It means splitting up the DNA somewhere and inserting DNA from another organism or something else into the original DNA. Then you will need to replicate that DNA because you want to replicate the cells. So you want the cells to divide so that you can produce lots of them. And once you have that extra piece of DNA in there, even an extra gene which might code for insulin production, you've maybe put it in something very simple like a bacterial genome, which is very easy to grow lots of in the laboratory. So you've spliced the genome of the bacteria, and you've spliced also spliced the gene out of a pancreas cell which produces insulin. You would then put that gene into the bacterial cell, and so when you do replicate the DNA this time and get your cells to divide, sorry, that was Timmy sneezing, Timmy the cat. When you do finally get your cells to divide, and this involves enzymes in the lab as well, those cells will then have an extra gene or a different gene, a gene which they didn't have originally. So this time, if you've inserted a gene that codes for insulin by splicing the genome, splicing your DNA, then each new cell that is produced will have the gene that codes for insulin production. So in that way it's going to produce lots and lots and lots of insulin. Every cell has the gene which is going to code for the production of insulin. So obviously bacteria don't normally do this, but often we use E. coli. You might know of E. coli as being an evil bacteria because it causes lots of diseases as well, but it's also helpful in our digestion of our food. E. coli is all over the place, and it has a very simple genome, so it's one of the bacterial cells which are used in gene splicing a lot to produce products that we need lots of, because you can produce lots and lots of a huge culture of this bacteria which has a gene spliced into it from something else, another piece of tissue, which codes for a product that you want. So every cell that is then produced by DNA replication is going to produce that product that you want, so that you're able to produce medicines for people. It could be insulin, it could be growth hormone, it could be something like a tomato plant that you're trying to produce resistance to disease. So you might take a plant which doesn't grow very well in your environment but is very resistant to a certain type of disease, and you're trying to culture some cells of tomato cells of a plant which does grow very well but is not resistant. So in this case you would splice the genome and you would take a gene from the tomato that has the resistance and you would insert it into your tomato genome that you want to produce resistance in, and then you would get the DNA to replicate so that each cell has a copy of that gene to make your plant that does grow in your environment resistant to that type of disease as well. So that's how it's used in disease resistance. Genetically modified organisms are, obviously the clue is in the term, so you're genetically modifying it. So if you're genetically modifying something, it means you're going to modify the genetic information, which means you need to split the genes again. You need to use an enzyme to split the DNA and to insert a different gene, which is probably from a different species in some cases. So I don't know if you've heard of the fishy tomato. It scares me a little bit, the fishy tomato, but it's found in the northern hemisphere, and in parts of Europe that are close to the North Pole it's quite difficult to grow tomatoes. So